Hello, good afternoon, good morning, good evening, everybody. I am here to get you started on the design flow experiments that are reproducible and third reviewer proof webinar. So thank you everybody for tuning in. My name is Kathy Sorbara and I'm part of the expert cytometry team. And I'll be with you for just a few minutes before I pass you over to our host, Tim Bushnell. So you're in the right place if you came to see the Expert Cytometry webinar on designing flow experiments that are reproducible and third reviewer proof. More specifically, you're in the right place if you want to learn Begley's rules for reproducibility and how they apply to flow cytometry. So if anyone's online now, if you can hear me and see my slides okay, it'd be great if you could type yes for me in the chat box, just so I know that our technology is doing what it should be. And we're going to get started at the top of the hour. Hi, Asia. Thank you. Good to know that everything's working well. And before we get started, it's always great to hear who is joining us and from where you are. So please enter your name and city or country into the chat box as well. We have a few minutes before we get started. And thank you for everyone for letting me know that my slides are all visible. That's great. So where is everybody joining us today? Sanjay from Pittsburgh, welcome. Great to have you with us. Anybody else? Where Krishendu, Jongho, Vanier, where are you joining us from today? And we are going to get started in just a few minutes. Calcutta, India, fantastic. Wow, Maputo, Mozambique, great to have you here. Bogdan, nice to see you again. I think I saw you on Monday's webinar. So thanks for joining us again, all the way from Romania. And Awara from Calgary, Canada, welcome. Great to have you with us. If you're just joining us, Asia from the land of wind and snow, great to have you here. If you're just joining us, you're in the right place to see the Expert Cytometry webinar on designing flow experiments that are reproducible and third reviewer proof. And we're just welcoming everybody into the webinar room now. And I'll give a brief introduction before I hand things over to Tim. Hi, Gia from Durham, North Carolina, and Keem from Bethesda, and Ellie from Cyprus. I think I also saw you on Monday, Ellie. Great to have you here. Laura from Missoula, Montana, welcome. And the good news is, just for showing up today, everyone will be able to get a bonus. So right now you can download Tim's advanced training on modern flow cytometry, which goes in depth on many of the topics you'll hear today. There's absolutely no replay of today's webinar, so you'll want to get the free bonus now. However, you can only get the bonus if you have a Facebook account. So set one up now if you don't have one. It only takes a few seconds. And to get the free guide, I'm going to put a link here in the chat box. And just give me a second. There it is. You should see it at the top of the chat box now. If you click on that, it will take you to this page. Once you're on this page, you just have to click the blue button that reads Send to Messenger and use Facebook Messenger to message Tim the word flow. As soon as you do, you'll get directions for downloading the guide sent directly to your Messenger account. And you'll have to do this now because once the webinar gets started, the bonus will be ending. Hello, Yorba Linda, California, USA. Great to have you here. Hi, Karen from Dublin, California. Thank you everybody for joining us. We are going to get started in just a few minutes. I'm going to give an introduction to Tim, our host for the evening. And I'll also let you know that towards the end of the webinar, you'll also have a chance to get a webinar only bonus. So this is a bonus that's available only to those who sign up for the mastery class during this webinar. So the webinar bonus is Tim's complete figure preparation course that will specifically show you how to create flow cytometry figures that reviewers want to see. So no matter what design software you use to create your scientific figures, be it Illustrator, Photoshop, or even just PowerPoint, this course will show you how to set your axes, how and where to add stats, as well as which stats to show, just as an example. So you don't want to miss out on this course as it has a $250 value and it's not offered anywhere else. So in fact, Mastery Class members don't even have access to it unless, of course, they became a member during this webinar. So it's definitely worth, if you're interested in joining the Mastery Class, to do so during this webinar. So once again, you're in the right place if you came to see the Expert Cytometry webinar on designing flow experiments that are reproducible and third reviewer proof. More specifically, you're in the right place if you want to learn Begley's rules for reproducibility and how they apply to flow cytometry. And we just have a couple more minutes before the start of the hour. If any of you are joining us today and just entering the room, you're more than welcome to type in the chat box where you're joining us from, because we love to hear where across the world we have people joining us. And we're going to start in just a couple minutes. And also, I mentioned previously that we have a bonus just for showing up to the webinar today, and that is this free video training on the 5 best practices of advanced flow cytometry. And in order to get this bonus, all you have to do is click on the link that I've posted to the top of the chat box, and it will take you to this page. And you just have to click this blue button on the bottom here that says Send to Messenger. And if you message Tim, he'll be able to set you up with this free bonus. Hi, James from Frederick. And hello, Jacob from Brooklyn, New York. Great to have you guys with us. And last but not least, we do have this webinar only bonus. So if you join the Mastery Class today during this webinar, you will get this bonus, which is Tim's complete figure preparation course. So it will specifically show you how to create the flow cytometry figures that reviewers want to see. So that's our special bonus for you today if you join the Mastery Class during the webinar. All right, so let's get started. As I said, you are here for the design flow experiments that are reproducible and third reviewer proof webinar. So our speaker today is Tim Bushnell, shown here. Tim did his postdoc at the University of Rochester and worked his way up to be the director of the flow cytometry facility at the University of Rochester Medical Center, shown here. He's been the director of this facility for over 15 years now. Recently, in 2012, Tim became the director of all URMC research cores, now titled the Shared Resource Facility Director. Tim specializes in flow cytometry, specifically the design of polychromatic panels, data analysis, quality control, instrumentation, small particle analysis, and much, much more. He has been an executive committee of ISAC, which is the International Society for the Advancement of Flow Cytometry, since 2008, having served as secretary and co-chair of the Membership Services Committee. In 2012, Tim saw a need to provide expert flow cytometry training to labs and scientists around the world, and so he funded Expert Cytometry, which specializes in training both academic and industry professionals in flow. Since founding Excite, Tim and his Excite team members have worked with the world's largest universities and institutions, including the world's largest biotech and biopharmaceutical companies. And this includes Bio-Rad Laboratories, BD Biosciences, Beckman Coulter, MedImmune, Thermo Fisher, Regeneron, Celgene, and many, many more. Excite's mission was to bring the best practices of flow to the scientist's doorstep. But Tim quickly realized that doing more than 40 live events a year around the world was not scalable and even possible for him. So as a result, Tim created the Flow Cytometry Mastery Class, a virtual flow cytometry training facility where scientists could get training in flow basics and where power users could stay up to date on flow best practices and new flow technologies. The Mastery Class now has hundreds of hours of flow cytometry training in everything from compensation to data analysis and statistics, to antibody titration, immunophenotyping, small particle analysis, proliferation and cell experiments, apoptosis, experimental design, panel design, and much more. Most recently, Tim added over 40 new high-definition training videos, or platinum content, as Tim likes to call it. These new materials showcase Tim's training in front of flow cytometers and cell sorters, advanced and interactive whiteboard trainings on core flow topics, and of course, a lot more. Okay, so that's enough about Tim and his flow exploits for now. But one last thing I wanted to mention is that Tim is also an incredible family man, volunteering a lot of his time to his kids and their adventures, including his son Nathan's Boy Scout troop. All right, so we're ready to bring Tim on and get started. Be sure to turn off your cell phones and put away your distractions. You can save your questions for the end because Tim will have plenty of time to answer them. So without further ado, here's Tim.
Good afternoon, and thanks for that introduction, Kathy. I laugh at that picture of the U of R.
Wave hello to everybody.
Oh, I hear.
Thank you.
It's snowing
Thank you for paying
attention. So let me get started. Turn over my slides. Where is that button? There we go. Out from around the world, and I appreciate you spending a little bit of time with me. Today we're going to talk about the concept of reproducibility and best practices in flow cytometry. And this is a highlight of a lot of the material that we cover that in a lot more detail in the mastery class. And at the end of the day, I'm hoping after you've attended this webinar, you're going to have a better understanding of some of the principles and practices of flow cytometry, or in flow cytometry, to generate and present high quality reproducible data. Now, reproducibility is not just a single panacea. There's no one thing that we have to do. It's kind of a mindset that we have to get into. So why are we really focusing and talking about reproducibility? Well, reproducibility is a central part of the scientific method, as this comic illustrates. But I'd like to highlight this article that was published in Forbes magazine, which Bruce Booth highlights a paper by Glenn Begley and Lee Ellis, where they cite 53 landmarks oncology studies, and only 6 of those could be robustly reproduced. And more importantly, as I showed, the irreproducible findings actually outpace those reproducible findings if you look at the number of citations. So that's kind of a concern. And Begley and Ionis and others have followed up on this work. And this is just a list of some papers from Begley and Ionis in the Circulation Research showing what concerns were in reproducibility. In fact, this paper cites a series of studies that estimate between about 75 to 90% of biomedical research is not reproducible. So if you think about that in dollars and cents, we're talking about 85 cents on the dollar. So only about 15 cents of the money we spend in science and research is actually considered reproducible. So this got everyone's attention, obviously, and NIH in the US has established a response to the reproducibility crisis, and they've added several requirements that you must report on as a scientist publishing or submitting a grant, an R01 or P9 or any of the other grants. And these include 4 very specific areas about the premise of the research, how the experiments were designed, consideration of different biological variables, and importantly, the authentication of key biological and chemical resources. So these are very broad-based topics. What Booth's article in Forbes kind of summarized out of the work of Begley and others was these 6 questions, or as he called them, Begley's rules. So when you're reviewing a paper, when you're reading a study, when you're looking at your own data, these are the types of questions you want to ask yourself to see if you feel that your data will meet that requirement, meet that mark of reproducibility. Now, the first question is, were the studies blinded? Were all the results shown? Were the experiments repeated? Were the positive and negative controls shown? Here we go. Were the reagents validated? And last but not least, were the statistical tests appropriate? So I'd like to take these 6 points and highlight briefly how we can address some of these concerns in flow cytometry and how you can quickly, with some additional forethought and additional planning, address these in your own research. So the first question is, were the studies blinded? Now, in clinical trials, that's relatively easy. The whole idea is that we're going to blind information about the test from the participant. So a patient may be getting the drug or the placebo. And in the clinical side, we do a whole series of tests, so we can do double-blind studies. So in fact, even the experimenters don't know what the patient's getting. Now, in the research world, when I was in the lab as a postdoc, we never did this. I would go down to the vivarium. I'd get the mice that I needed for my experiments. I knew what the mice were. I'd extract the tissues. I'd label them in, control in one tube, experimental in the other. And so there was an inherent potential to bias the results because I knew what was going on. At a minimum, I would encourage you to get your lab mate to go down to the vivarium. Or better, if you get the vivarium and bring the mice up, give them to your lab mate and let him give them a code or a number. And then you only work with that number, and you don't know which mouse is which until after the experiment's done. That way, you have a less chance of biasing the results, just even without being conscious of that. So it's a very good, simple way to improve your work and improve that the studies you're doing are blinded. It's even easier if you're getting blood from a lab. You can just, again, have somebody label the tubes and give a code. Of course, the second important question that was asked is, were all the results shown? And how do we think about that? Well, first off, in current publications, a lot of information is not available. And this can include things like the specimen information. Staining conditions are not always clear. I've read a lot of protocols that will just say, stained using standard protocols. Well, which standard protocol are you using? Because time and temperature can affect antibody binding. The rate of antibody binding at 37 degrees or room temperature is very different than the rate of antibody binding at on ice at 4 degrees. Letting the antibody binding go for a half an hour versus an hour can also have a big effect. Second thing that can have a huge effect is what buffers you're using. And this is especially true with intracellular work. The composition of the buffer can impact both the ability of the reagents to bind as well as the fluorochromes attached to them. So if you were to surface stain cells and have, say PE and FITC on the surface and then treat the cells with ethanol, you're probably going to kill your FITC signal pretty quickly because FITC doesn't like to be ethanol fixed. Antibody information is often complete or lacking. Since clones have different affinity, it doesn't help if you say, we use clone CD3 from Bio-Rad or, BD. There's 5 different CD3 clones. Which one did you use? Likewise, you need to make sure you can indicate what fluorochrome that you use, because fluorochrome choices will have an effect on the sensitivity. If you have an instrument and you've put trying to make a sensitive measurement in the PE channel and you have a lot of tandem dyes that might spill over into that channel, that could compromise your data. And oftentimes people will just say, data was acquired on a, BD LSR2 or a Beckman Coulter Cytoflex without giving information about laser power, laser configuration, filter configuration, and all of those are going to have an impact on the sensitivity of your measurement and should be addressed. Now, there is a solution around this. This is called the MyFlowCyte, or it's MyFlowCyte compliance, and it's the minimal information about a flow cytometry experiment. The standard was approved by the ISAC membership, and the reference for this, if you're interested, is Cytometry A here in 2008. Currently, it is used by in the journal Cytometry A and the Nature publishing groups. And if you provide this information, your paper gets a tag that indicates that it's MyFlowSite compliant. Ryan Brinkman, who was the head of the ISAC data task force that established this, he'll actually be speaking into the mastery class at the end of the month about automated data analysis. But you can, this is an important component of that as well. Some of the information that you include: experimental overview, the flow samples. Notice we're going to talk about the specimens and the treatments. This is going to answer that second question. What do we know about the specimens? We're going to talk about the data analysis. You can see you're going to talk about provide the list mode data, discuss the principles, how you compensate it, how you gate it, and what statistics you extracted, and the details. So this MyFlowSite compliance really helps answer a lot of the questions that Begley's rules highlight. Likewise, the question is, was all the data shown? Now, it's not always easy because you might do 20 experiments and you're showing the statistics on those 20 experiments. However, the Flow repository, which was started in conjunction with ISAC and a grant from the Wallace Coulter Foundation, is a place for flow cytometry researchers to put their data so that others can access it. Obviously, they have a moratorium system so that it can't be accessed during review of papers. But once your paper's published, people can go in there and pull out your data. And if you've shown how you analyze something, they can test and check the analysis as well to confirm and see what you were seeing. And as new methods come around, we can reanalyze this data in a variety of ways. In fact, a paper published early last year on a program called FlowClean went into the data repository and analyzed 13,000 different files to look at how well the stability of the instruments was on these files. And they found in roughly 13% there are some stability issues. So it became a useful tool for this program to test and see how it could find in a wide variety of data sets. So let's talk a bit about, were the experiments repeated? Now we're going to get into a little bit of detail here, and I don't want to scare you off about statistics and things, but reproducibility requires us to do multiple samples. You can't just run one sample and say, oh, I found something. There's a great YouTube video joke about this. If you search biostatistician and researcher, where the researcher keeps asking if he can only use three patients. We think of this as powering the experiment. And the concept behind the power of an experiment is a measure of the fraction of experiments that would be expected to yield a significant p-value. The greater the power of our experiment, the greater the chance of reaching statistical significance. We define this as a beta, which is here, 1 minus the power of the experiment. It's a little bit confusing, but we'll talk about that later. And it's a measure of making a type 2 statistical error. Now, how do we power our experiments? We do something called the power calculation. And this is a statistical method to prospectively determine how many samples should be used in the experiment. Again, we typically will set this power at 80%, but you can set it at whatever value you want. Now, if you set it at 50% or below, it's probably not worth doing the experiment. So you really want to at least hit that, I believe, hit that 80%, if not higher. Now, if you haven't done this and you go ahead and you gangbusters and do your experiments and you don't find significance in your data, you can do a retrospective calculation that will determine if the effect measured was too small to be discovered because of your sample size. So rather than do it retrospectively, do it prospectively and know what you're going to get going into it. Now, there's a lot of ways to do this. There's a lot of statistical programs out there for various analysis tools. I happen to like this program, StatMate Analysis, and it's published by a company called GraphPad. And for you who are Mac users, you can't use it on a Mac unless you've got an older generation one. It's a PC-only package. What this does is if you put into the software the type of test you're going to do, the statistical test, a t-test and ANOVA, you put in your threshold value, your, the 95, your confidence level, you're going to test at an alpha of 0.05, 0.001, whatever. And something about the distributions of the data, in fact, the spread of the data, and you can get that as an estimate based upon the literature or other information you have. That information gets put into the software. The algorithm then spits out this table. Across the top are different power levels from the 80th percentile up to the 99th. Down the side here is an N. This is the number of samples per group that you need to measure. And cross-referencing the number of samples with the power will give you a value down here. So what are these values? These values are estimates based upon the difference between the control and the experimental population you're trying to find. So in this case, if I were to say the untreated fluorescence intensity of my sample was an average of 3,500, and I wanted to find a 10% difference, that would mean I want to find a difference that's 350 fluorescent values above that mean, for example. So how many samples am I going to need to find this difference? I cross-reference this number, 350, in this area with the power of my experiment, and then I will figure out the number of samples. So if I want to power this at the 99th percent level, you can see here I've boxed, we're going to need to do between 20 and 25 samples per group. If we want to power it at the 95th level, we're going to need to do, somewhere between 18 samples, between 16 and 20. If I want to power it at a lower power, coming down here to the 90th percentile, you can see we only need to do about 9 samples to power it at that level. I'm sorry, 350. It's right there, 16, I'm sorry. If I want to find a bigger difference, say at 20%, now I'm at 700. You can see 700, I need to do between 5 and 6 samples to be powered at the 95th percentile. So the trade-off here is the bigger the difference between the control and the experimental, the fewer the number of samples we have to run. The smaller the difference, the more samples we're going to have to run. So this chart helps you determine how many samples you're going to need to run and give you a choice so you can understand the consequences of what that choice would be. The higher the power, the greater chance of finding significance, but also the more number of samples you're going to have. So I'll just briefly mention again, there's that webinar bonus that's going to be coming up. You'll see a green window pop up in your chat window in a little bit, and that is a webinar only bonus. You have to sign up with a mastery class today during this webinar to get that bonus. And again, this information I'm presenting today is just a sampling of the hundred of hours of videos that we have, and the where we continue to add between 20 and 30 hours worth of new videos every year. There it is, the mastery.expertcytometry.com. If you go to that site and sign up, you'll get that special webinar bonus. So now, while you're looking at that, let's talk about where the positive and negative controls shown. This is another one of Begley's rules, and it's a very important one. And let's talk about the types of controls that people will use and which ones you should show and which ones you should avoid. The first one that I'm going to point out is something called the isotype control. And I'm sure you've seen this in papers and you've heard about it. The concept being that all antibodies have a specific isotype, typically represented by the heavy chain and the light chain. So for example, you might have an antibody that's IgG2 kappa light chain. That will then have a specific FAB fragment that may be binding to the CD3 molecule on the surface of a cell. Now, if I take an antibody with the same isotype, but to an irrelevant target, the theory is that if I use that isotype control, I can determine the positivity of the sample because I'm going to be able to set and determine the nonspecific binding of my antibody. So these have been used for many years, and the problem is that people use them without thinking about them. The assumption behind the isotype control includes the affinity of that variable region of the isotype control has the same characteristics for the secondary target as the primary antibody. So the question is, how do you know that? We don't. Second question, or second assumption, there are no primary targets for the isotype antibody to bind to. And do you actually know what the primary isotype antibody is? There's a very common isotype control sold. The MOBC is the clone name. This clone was first published in the mid-1970s. And in those papers in the 70s, they specifically said they did not know what the target of this clone was. If you read the vendor literature, you'll notice that the vendors say that they've tested this clone and shown that it has no binding against common major subsets. So they've stained CD3 cells and they don't see anything major. They see stained CD4 cells and don't see anything major. But who knows if that's really the case? If we don't know what the target is, how do we know that we haven't excluded the target because we've assumed that the isotype control was measuring nonspecific binding? Another factor that we assume is that the fluorochrome to protein ratio is the same. In the case of, say, PE or APC, the very big fluorescent molecules, that's probably a reasonable assumption because those will be at a 1-to-1. But if you're looking at FITC or Alexa 488 or one of those dyes, those are smaller. And they can have very different F to P ratios based upon a variety of factors about the antibody. So as I said, the historical practice was this was used to determine positivity. The actual use is basically only to determine if your blocking was efficient and effective. Now, while some kits contain matched isotype controls, there may be some value in them as in intracellular staining, but especially on surface staining, you should avoid them. Not spend the money, use the appropriate controls to set positivity. And here's two pieces of data just to kind of highlight that. This is from this paper, Macker and Trotter, 2006. They have unstained, as I said, gated on small lymphocytes, and single stains with IgG2b phycoerythrin, IgG1-PE, IgG2A-PE. This red line is mine, kind of showing you the mean of this peak down here of the unstained. And you can see each of these isotype controls have very different binding characteristics. So we don't know what they're binding to. We just see that they're shifting. We don't know what's causing that shift. More convincingly, I believe, was this paper published just in 2006 by Anderson and coworkers. And what they showed is they were looking at the best way to block human monocytes and macrophages. And they know in the cells that they were studying that these cells express the Tie2 antigen on the surface of their cells. It's expressed at relatively low levels, and so they stained surface staining for those, and you can see here the background staining, and then the positive staining shown here. And you can see there's a shift. It's small, but there's a positive shift to that. They then went and stained with an isotype control from a vendor, and look at where the isotype control showed up. This was well over a log higher than the actual signal. If we followed the idea of the isotype controls, we would say that these cells were negative for Tie2, when in fact we know they are. The paper goes on to say that they got isotype controls of IgG1 isotype controls with Alexa 647 from different lots, and the binding of the isotype controls was very different and very lot-to-lot specific. So if we don't even know how the lot-to-lot variability is going to affect the binding, how can we rely on this as any level of control? Now, if you are subscribed to our Expert Cytometry, the Xcyte Facebook page, you follow us on LinkedIn, or you follow us on Twitter, we put up a series of essential references that all flow cytometrists should have. And as one of those, we put in a series of references, about 5 or 6 papers that talk about this idea of the isotype control. So if you're not convinced by today's lecture and this specific example, there's a bunch of papers that are out there that will hopefully help convince you, and you should move away from this control. So you can ask, what should I use instead? How do I set positivity? Well, one control that you should use is the FMO control, the fluorescence -1 control. This is a true gain control. It identifies and addresses the spread of the data. And what it's going to do is it's going to reveal the contribution of error measurements in the channel of interest from all the other fluorochromes in the panel. The use of the fluorescence -1 control is essential when you're looking for accurate discrimination of rare events, when antigen expression is low or variable, when you're looking for the emergent appearance of a antibody or an antigen, such as an activation marker. When I'm developing a polychromatic flow panel, as I'm going through the validation steps, I will run every FMO control. The goal of running every control at the beginning of the experiment is to determine which controls are essential for me to properly gate and identify the populations of interest. Those controls that do not add value to that process, I won't carry forward into the production, when I'm actually running the assay for real. So, for example, it's unlikely that I would use a viable or a live dead or a dump channel FMO. Likewise, I probably wouldn't use a CD3 or CD4. But if I'm looking at a rare population like activation marker CD86 or the expression of IL-2 on stimulated cells where this can be relatively low frequency, I am definitely going to be using the FMO control. You may have seen this before, but just in case you haven't, here's what the FMO control is and how we use this. So we've a 5-color staining panel, FITSI PE, PE 5.5, as well as APC and DAPI. Now, if we look at a completely unstained sample shown here on the left, PE versus FITSI, this is what our signal looks like. Historically, we draw a line from that, shown here in red, and say things above this red line are positive, things below are negative. So on the fully stained sample, okay, these are double positive, these are really positive. That's how you would interpret it. But look at this plot in the middle. Again, PE versus FITSI. Notice these cells in the middle have no PE staining. So how do you interpret these populations that are above the red line based upon the unstained boundary? We know that they can't be PE positive because there is no PE in the sample. Therefore, the FMO control lets us say that these cells here, this staining, is due to contribution of error to the measurement of all the other fluorochromes spilling into the detector of interest. So the true gate is shown here by this blue line, the FMO control, FMO bound. This delta between the red and the blue line is often referred to as the spread of the data. And this is a spread caused by using the fluorochromes on your cells and seeing how they affect the fluorescence in a specific channel. As the number of colors increases, the spread can get wider. Now here is a 12-color panel, and here we're showing Texas Red PE and Alexa Fluor 700. This is off the green laser, this is off the red laser. Excuse me, here's our unstained bound in red. The middle panel is our FMO control. That is the blue line there. This difference here is a spread of the data. Now here's the green box. These cells in the green box are cells that we would carry forward because we know that they are above the background in our sample. These red cells in this red box here, we're not quite sure what they should be. Should we carry them forward or not? Because by the FMO control, they tell us that they're within a range that they could be confused or confounded because of the spread of the data. These could be bright negatives. These could be dim positives. And so you will have to make a decision as to where your cutoff is. If you have, in this case, this is a very strict cutoff. If you have additional fluorochromes, additional antibodies down, you may carry on and bring these forward, this smaller level. As I said, not everybody uses a CD4 isotype control. but you want to use this to identify, and you're very confident that these cells are the cells that should be looking at. Another thing that really is useful when you're looking at your developmental work as you're developing your assay is trying to identify internal negative controls. The idea is that you are looking for a cell population that is similar to your population of interest, that you can easily find with the staining parameters you're using, that you know is negative for the target reagent. In this case, we're looking at CD4 cells versus an activation marker. We've identified a population of cells that are CD4 negative but are positive in other areas, and they're also negative for the activation marker. So that allows us to establish a range of expression. Here's my negative control. I know up to this point here, this vertical line, these cells should be negative and are negative. So that helps me say that these cells are positive, and these cells, I'm not quite sure what they are. They're in the negative. They could be dim positives. They could be floating, but these cells are truly positive. It's not always easy to find one of these, but if you can, it's a really useful tool. And it's what you look for as you're developing your assay. Any place where you feel, again, rare events, emergent dyes, etc., where you're going to be able to get better resolution by having these, you should use them. Continuing on, another important control is the idea of the unstimulated control. So the unstimulated control is used whenever you're doing a stimulation experiment. This lets you take advantage of the biology of the system to help you further set your gates. On the top panel, we're showing SEB-stimulated T cells, and we're looking at expression of CD4 versus IL-2. We know that IL-2 should not be positive. That should not be positive on unstimulated cells. So let's look at the three types of controls that we have. Here in the middle, we have our FMO control. The FMO control is the first control that we use that helps us set positivity from negativity based upon the spread of the data into the channel of interest. So we might want to put our gate much closer here. The second control we have is the isotype control. Now, as I said before, that's not a good control to set positivity, so we're going to ignore that for a minute, and I'll show you the consequences of that isotype control. Over here, we have an unstimulated control. These are the cells that have been fully stained but have not undergone the stimulation, the SEB treatment. So we're going to take advantage of the biology, knowing that these cells do not express IL-2, and we're going to set our gate. And that's the gate that's shown here. When we're setting gates based upon a control like this, like an isotype or an FMO control or an unstimulated control, I like to observe the rule of statistics that I want my population in this region here, a gate, positive gate in the FMO or unstimulated control, to be less than 0.1% of the population. That comes from the idea that 99.7% of the data is within 3 standard deviations of the mean of that data. So that means roughly 0.15% of the data is above that 3 standard deviations and 0.15 is below. So I've pushed it out a little bit further to say less than 0.1% of the population, positive population, is in the gate. That's going to help me account for any schmutz or things that I may have missed. Of course, I will go back gate and make sure I didn't obviously screw up somewhere in my gating. But once I set that, then I'm comfortable with where I've placed everything. Now, quality control is a really important topic. That's something that lets you know that the machine is working, something that people who are running your instruments, your core managers, facilities directors, whoever they are very passionate about and working very hard at. And that idea is that we track and know how the instrument is behaving every day, so that when you come into the facility, you know that it is behaving. And when we did QC, everything was well. Now, of course, over the course of a day, QC, instruments can get banged around. And when machines get banged around or they get cluttered up because of people not taking care of them, that can affect the data. One way to address that is this concept of a performance tracking using a reference control. A reference control is a sample that you know the behavior of that is reproducible in your assay. In this case, we had access to hemochromatosis patients, so we were able to isolate large numbers of PBMCs and freeze them down. Every time we ran this assay, we'd pull we'd pull a vial out, we'd thaw it, we'd stain it, and we'd measure it in the assay. And as long as it was within the range of the assay, the acceptance criteria, we would move forward. In the case, like, for example, here, this little plot, that it was not, then we would immediately start to do some interventions, start to try to troubleshoot what might be causing that problem. This benefits you, the researcher, by giving you an additional level of confidence without having to learn a whole new protocol and then track somebody down for the beads and run all that you can use and track. The next concept that Begley asked was, were the reagents validated? Now, this is a really big issue. If you're familiar with the concept of cell line drift, you know that cell lines can drift over multiple passages. You know, contamination of cell lines is a common occurrence if you're not really careful. So how do you know that the cell line that you're using is actually the cell line that it says it is on the tube? Well, right now you can do some sort of genetic screening using a PCR-based method, or just sequence the bacteria or the cell line to make sure that it's what you think it is. But what other reagents should you validate? Well, I'm going to argue for a minute that the instrument itself is a reagent. It's a measurement tool that's going to help you determine positivity, negativity, and gather data. So we want to make sure the instrument's characterized and properly QC'd at a different level. Antibodies are reagents. We're going to want to make sure they're properly titrated and are being used at a proper concentration, that they have the right fluorochromes on them. The cells, we want to make sure that we have proper preparation of that. And of course, ultimately, this builds up to validation of the experimental design, showing that the experimental design is correct and that all the pieces downstream have been validated to get to that point. So quality control is ensures consistency and reproducibility, and this includes measurements of sensitivity such as PMT sensitivity, laser alignment, stability of the fluidics in the background. This can be optimized for each facility. You need to learn your instrument, how the instrument behaves, and go ahead and ask the core facilities how they are doing their QC. You know, feel free to ask them what the QC looks like. I'll tell you, here at our facility, we dump everything into a database program automatically that's automatically analyzing all of our QC data. And we have QC on every instrument that we've had in our facility and every PMT in our facility since we put this program in place in 2008. So that's, you know, almost 10 years worth of data. And I can tell you, if you give me a day of the week and ask for your machine, I'll tell you how well that machine was performing in relative to its baseline. That gives the user base a level of confidence that the machines are working consistently and reproducibly. One of the ways we do that is we optimize the PMTs. Old school voltage setting required you to put a unstained cell on the instrument and adjust the voltage till the cells were in the first log decade, down in the little lower left quadrant. With the advent of digital instruments, it became easier for us to actually optimize the PMTs and become independent of arbitrarily setting the voltage based upon where the negatives fell. Now, if you have a machine, a BD machine, they do their CS&T, and that is actually will calculate that. But if you have a non-BD machine, or you're like me, and I want to have a second method to validate that. In fact, I only use CS&T as a useful for quality control tracking because it's measuring over consistency over time relative to itself. We optimize our PMTs using this method, the Peak 2 method. In this method, we run a dim particle, in this case, the second peak of the Spherotech 8-peak bead set, and we run that over a voltage range from, in this case, 200 to 900 volts. We identify the single beads, and we get the CV, the robust CV of the population. The robust CV being a measure of the spread of the data. That is the robust standard deviation divided by the median. You can see at low voltages, this CV is very, very high. This means the spread of the data is very wide. As we increase the voltage, the CVs start to drop. They go down and they hit an inflection point. And an inflection point, for those of you who remember your math, is a place where the curve, the slope of the line of a curve changes. So you can see we go from this to this. At high voltage up here, the signal isn't improving. That CV hasn't changed. So our optimal voltage is right just past the inflection point. We usually quote it within this 50 volt range. And this is a place that you want to start. When you come to the instrument, you take that standard voltage, you set your PMTs to that voltage, and you start to run your cells. And you don't worry about where your cells are on the axis. If you, for example, were to take your cells and put them on the instrument, and you saw off the red excitation in an APC-like emission, a 660-20, that the cells were off-scale negative, and you start to bring them up, all you're doing is adding error to your measurement. We run this Peak 2 method every 6 months, or every time something major happens to the optical pathway. A new PMT, a new laser, a new filter, a significant alignment difference. We publish these on our website so people know where to start. The other thing that you can calculate using that same bead set and just looking at the math slightly differently is the standard deviation of electronic noise. The concept here is that there's a background noise in all PMTs. This comes from dark current. This comes from random emissions of a electron off a dynode. It could occur because of radiation, ionizing radiation. It could occur because of leaking of light into the PMT. We want to find a place where the contribution of electronic noise is minimal. And so by knowing what the standard deviation of electronic noise is, we can do that. So here we've taken those same beads and we've plotted, in this case, 1 over the median squared versus the CV squared. That is then fit to a regression line. Here's the equation of that line. The R squared is 0.9989, a pretty good regression. It turns out the slope of the line is the variance in the data. If we take the square root of the variance, we get the standard deviation. So now we know the standard deviation of this is 18.59. When I put my positive sample on my unstained cells, I am going to raise the voltage until the standard deviation of my population in the negative region is 185, 186. So it's 10 times above that. In fact, this is what the BDCS&T effectively does to set the voltages that they publish in their baseline. Here's just a quick sample showing you the issues with laser alignment. Changing the alignment here on 2 different days. These black bars show the bounds of the population. A realignment shows, you can see some white space there. We've gotten that population tighter. The consequences of that are shown here. Here's the line for day 1. You can see by tightening up the alignment, improving the alignment, we get a tighter population. Then we gain about half a log of sensitivity. On those lasers that have what we call the parallel or sequential arrangement, the system has electronics that will delay the signal from each laser some unit of time so that when the delayed signals are all stitched together, they all represent the same signal from one cell. And that's what gets stored in the FCS file. Here you can see, for example, The laser delay between 488 and 405 is 20 microseconds, and between 488 and 640 is about 40 microseconds. So we need to delay this signal by 40 microseconds and this signal by 20 microseconds using our delay electronics so that the delayed signal is shown here. And that ultimately these three are stitched together. When that goes awry, you will see issues like this. This is what good stable flow looks like. Here's what bad flow looks like. In this case, we lost the signal. This was due, in fact, to a clog in the system, and the clog in the system can either cause the system to speed up or to slow down. In either case, it will miss that window that the system is looking for where the data should be. I recommend, if you've got a multi-laser system, to put up a plot of fluorescence versus a time for each laser using the fluorochrome that is the brightest on that sample so you can actually see the fluctuations. And that way, when you don't walk away from the machine, you can solve this before it happens. Of course, beads are good for quality control tracking. This is a 6th peak of the 8-peak bead set. We run this over every time we turn the system on, in this case, and we plot the data for each peak on a table like this. And this is a Levy-Jennings plot showing the average in blue and 2 standard deviations above and below the mean. So I can make a quick assessment of how far off from the mean are the populations of interest. Or I'm sorry, how far off the mean is the voltage, and is it a cause to worry? So you can see here, 2 standard deviations represents about 620 to 660 with a median of 640. So it's a pretty tight signal. If you are going to be using beads, and I encourage the use of beads, in fact, I'm writing a blog post calling a few of my favorite beads, you need to make sure to remember to do an overlap experiment. Anytime you're running out of the old vial of beads, you should run in parallel. I recommend 5 times at a minimum, the old beads at their target value and the new beads at the same voltage to figure out what the target value for the new beads should be. And then you record those target values, and that you replace the target values on your experiment based upon this overlap experiment, and you have traceability back to the original experiment. Are the reagents antibodies? This paper from Bradbury and Puckthorne and 110 colleagues who signed it illustrated in this figure very succinctly that in the US alone, this is a worldwide spending, about $1.6 billion in worldwide are spent on antibodies, about $700 million in the US, and almost half of that is spent on reagents that don't work. They don't bind to the right target, they're mislabeled, etc. So we need to ensure that our antibodies work. We want to make sure that the fluorochrome isn't binding. So if you haven't seen this paper and you're working with CD64 positive cells and you're using PE-Cy5, you could be having issues. This just shows it how it, this paper shows it how it binds. Now, to determine if that is happening, you can do the isoclonal binding experiment. And this is where you take unlabeled antibodies that are the same as the ones that are conjugated, and then you start to mix them in a 1-to-1 or 1-to-4 ratio. Now, if the binding is specific and it is the antibody, this will titrate down. As you can see, as I doubled the concentration, my staining index went down. If this is binding to the fluorochrome, this isn't going to happen. You're going to see the same thing here. So if you're using new reagents that you've never used before, this is a useful tool to confirm to yourself that the fluorochromes don't bind to the antibody or don't bind to the cells. Let's round out our talk with the last question that Begley asked, which is, are the statistical tests appropriate? Well, the steps in hypothesis testing are very straightforward. And these are the things, these first 2, that you should do when you're designing your experiment. You should establish the null hypothesis, and you should set the threshold value. Once you've done that, then you can go ahead and proceed to do your experimentation, because After that, you're going to take these hypotheses and the tests you've decided on and perform the experiment. That threshold is historically set at 0.05. This is based upon the idea that, or is interpreted as there's a 5% chance of finding significance where there is none. That is the idea of committing a false positive or a type 1 statistical error. As you increase the threshold, it becomes easier to find significance. As you lower the threshold, it becomes harder to find significance. Now, this 0.05 number came out of a paper in the 20s by R.A. Fisher, one of the founders of modern statistics. Unfortunately, people saw that one number and took it as the be-all and end-all, when in fact Fisher went on to say and reiterate in 56 that this alpha needs to be set based upon the conditions of the experiment and not should be moved based upon those considerations, as well as considerations of the consequences of committing a type 1 or type 2 error. If you don't know what test to choose, this is a good chart to work with. I have found myself that most flow experiments break down into some very straightforward questions that usually result in either doing some sort of t-test or based analysis of variance test. But we're going to measure that based upon what we're trying to describe and how the population is distributed. The ANOVA test is important to remember if you're doing more than 2 pairwise comparisons, because if you do just 2 t-tests back-to-back, sequential t-tests, you'll see that you have roughly a 10% chance of finding significance where there is none. And this goes up rather rapidly. So if you do 4 pairwise comparisons, you've got almost a 20% chance of finding significance where there isn't any, committing your type 2 errors. As I keep talking about what is a type 2, type 1 error, type 2 1 error is a true null hypothesis is incorrectly rejected. That's the false positive. The false negative, the type 2 error, is related to the power of the equation. And this is where a false null hypothesis was not rejected. So, in summary, to improve reproducibility, you must be open to a new mindset on how you do your experiments. And you can improve this by doing some simple things like optimizing the quality control on your instrument, the quality control on your reagents, the quality control on the assay, so you have that data and you can show the consistency. You can improve this by powering the experiments correctly, and improve this by performing the correct statistical tests while minimizing type 1 and type 2 errors. And last but not least, by reporting everything. Take advantage of the MyFlowSite standard, take advantage of the Flow repository so that people have access to your data and can look at that and look at the conclusions you drew off the same data. So can you do this? As you probably are aware, we're in the middle of our winter enrollment with the mastery class. And the mastery class, as Kathy told you, came out of the need and the desire to bring the best practices to researchers like yourself. And since I can't be in all places all the time, being on the road for 40 lectures over the course of a year or so gets very tiresome. So we felt, and it's not always easy to get everyone to the room at the same time. So the idea behind the mastery class is this is a online self-paced training program. And we're going to help you address issues like this. The estimate for irreproducibility based upon the empirical observations ranges from 75 to 90%. And roughly 85% of the data is proportional for biomedical research is wasted. And this irreproducibility is seen across the spectrum of biomedical research. And again, this is a Bradbury issue about the reliability of antibodies. So this is what you get in our program. You get a set of core modules. So if you're a new user, these core modules will get you up to speed with the fundamentals. If you're a seasoned user, these are good things to review. There's tips and pointers that you may not have learned or may have forgotten in your training. We have quizzes to help test how well you retain that information. You get access to some control books for these instructional modules. And we have well over 100 different training videos on a whole variety of topics, as you can see here, from compensation and statistics and panel design to data analysis, FCS Express, to techniques such as fluorescent proteins and apoptosis and controls. We have about 20 to 30 new seminars a year, roughly one every 2 weeks. And as a member of the mastery class, you get a season pass to those webinars. This spring coming up, as I said, Ryan Brinkman will be talking about automated data analysis at the end of February. We have a talk on a new tool for helping you determine the best antibody clones to use from a company called BenchSci. We have a talk by statistician Regina Nuzio, who is a leading communicator in the field of statistics, and we'll be talking about the fundamentals of statistics. She's very well versed, very good at communicating. She's writes not only for the scientific journals, but is often found writing for some of the common regular, average publications, normal publications. You also get access to this private group. This is a group we use a Facebook interface. We've tried the Google group interface, but the Facebook is a lot easier, and most people have a Facebook. If you're like me, I only have a personal Facebook group just to watch my granddaughter grow up. But here in the private group, you get access to other members of the mastery class. You get access to myself and other experts. We share ideas. We talk about problems and solutions. We talk about papers that have been published, additional training. I do a lot of additional training there. We did a 3-week session in the mastery class private group on getting people ready to take the certification cytometry exam. And everybody who's run through our program has reported back to me that they've been able to pass, and that has prepared them well for it. I'm on that group multiple times a day and always ready to lend a hand. It's always great if someone has a member of the master class emails me a private question, I ask them to put it on the group so we can all talk about it. These are some of the highlights of our new Platinum content. This is the next iteration of where we're going. We're adding more of this information, more of these shorter videos. The webinars are 45 minutes to an hour. That's a long time to devote. These videos, these Platinum videos are 3 to 10 minutes on a focused topic. So just a very specific component. You want to talk about antibody titration, we spend 5 minutes, 6 minutes talking just about antibody titration. And so you're able to focus on those topics. If you need, there's something specific I need, you can go to one of these and you'll see it, either me teaching it on the whiteboard, teaching it in front of the instrument, doing screen capture technology. So we're actually now being able to record videos showing you what button to push on the software to get what response. I'm working to schedule another session in front of the whiteboard with our photographer, and hopefully when it's a little bit warmer than it is today. Here's just a highlight of our private group. You can see this, right? We had over 350 members when we took this picture a little bit ago, and I think we're almost at 400 members now. We should change that 200 members, annually. It's always growing for members who've joined. As you can see here, Pam asked a question about titration, and you can see within hours we had a bunch of people responding. They even beat me to the punch to talk about the way to calculate titration, measuring the staining index. The group is very user-friendly, very people-friendly. No question is ignored. It's always fun to get tips. I'm trying to sort these cells. I've never used them before. How do I do it? I'm trying to stain these cells. I saw this. What happens? And it's great to really give you the answer. You may have heard of the Purdue list. You may even be on the Purdue list. You may have heard of the cytometry Google group. You may even be on that group. Those each have their strengths and weaknesses. This is just a kind of a real-time chance for you to talk to people. These are some commentaries. You can see these on our website. If you go to mastery.expertcytometry.com, we have a bunch of testimonials of people who have been members of the program. And they range from senior core managers, such as Chris Groves, to researchers and postdocs like Rush here, and experienced beginners all alike, finding value in the program. If you join, you're going to see this. You'll get login credentials to this specific area. This is our Mastery Class dashboard, and you can see the different access. Here are our 4 modules, advanced videos, our webinars, and of course, platinum content. And you can see all that different platinum content, and you're able to watch these at your leisure. Works on the cell phone, works on your iPad, works on your computer. So today, this week, we have a special offer. If you join, it's going to be $385 on a one-time payment. That is equivalent to basically buying 1 antibody. So you're getting hundreds of hours worth of information that you might have to go to multiple scientific meetings to get, read and digest tons of papers for the cost of an antibody to improve your research, improve the consistency and the reproducibility of your data. Like I said, all the material that you will see in expert cytometry has been, discussed and collaborated with colleagues around the country, around the world, focused on finding the best practices and providing those to you. And it's independent of the instrument. We have specific talks on specific instruments, but the principles of data analysis, the principles of panel design, the principles of how the system works are all universal. And today, if you sign up, in the next few minutes, I'll answer a couple of questions before I have to go, but you'll get this advanced figure prep course. This is a course that talks about how do you analyze and how do you develop your figures so that you are comfortable in presenting your data. And you can see there are 4 videos, 4 hours worth of training, talking about different topics, from gating strategies to statistics and showing the reproducibility. So this is a website you want to go to. Just remember to sign up before the end of the webinar to get that special bonus. So let me answer a couple of questions. Toei asks, I hope I said your name right, and I think he's talking about spectral spread. Does this happen even with compensation in place? So compensation corrects for the spread of the data based upon the fluorochromes. What you're seeing with spectral spillover is related to photon counting statistics and the efficiency of counting photons and the error inherent in counting photons, which is independent of the compensation. So that's where that comes, and that's why we use it. Next question is from Verna. Vania. Again, I apologize if I said your name wrong. How to define the negative range on an X and Y axis in a quadrant graphic. The lines are fixed as a 10 to the third is a standard rule. Well, first off, that's not the case. It depends on the software you're using. Third-party software like FCS Express does not fix them at that. They may pop up in one region, but you can adjust them. And go in to set the gates up. More importantly, I'm not a huge fan of quadrant gates. I really do not use them for my work because especially because if you start to look at the data, the spread of the data is not in a nice uniform process. If you hear one of our compensation lectures, we get into the details about that. Verena asks about multivariate analysis, MANOVA testing, and that's a process for comparing the multivariate sample means, and this is when there are 2 or more dependent variables. Most of the work that I've done breaks down into just a typical ANOVA. I haven't seen a lot of need where I'm going to have multiple variables in that case. Again, it depends on your question, and so if your question is going to go down that route, then you're going to have to go in that process. So it all is going to depend on what your question is and how you're going to have to answer that. Dina asked, what is the explanation when you don't see differences in your titered antibodies? Your flow plots look the same with different titers. The cells were blocked with 10% human antibody serum. So the first thing you want to do is put those on I prefer bivariate plots. I don't like to look at histograms, and I'm going to calculate what's called the staining index or the separation index. And this is going to be measure the spread of the data corrected for the background. So if you do that and you still don't see anything at any titration level, there may be a problem with the antibody, or there may be some other issues going on, because you would expect to see, as you decrease concentration, that the antibody signal starts to fall off because you don't saturate. And at high concentrations, you expect to see the antibody signal of the separation index decrease because the spread of the data of the negative population starts to increase. So there's a question here, Asila, about taking the mastery class. I'm going to refer that to one of our, I'm not quite sure what you're asking. But I think personally, I think the program benefits the people who take it and take advantage of it. And if you have more specific questions, shoot us an email, expert@expertcytometry.com, and we can answer those questions for you or for your director as well. I see it's about 10 minutes after the hour. I'd like to thank you for coming to this webinar. I know we have one more tomorrow afternoon. Going to be talking about hacks to improve your flow analysis, as I recall. I have to look at my my specific topics. Thank you, Aja, for the answer. I think you understand the question better than I do. My mind's in lecture mode, so forgive me. So again, last couple of seconds, if you sign up for the mastery class at mastery.expertcytometry.com for that special $385 price, or $95 a month for 6 equal payments, then you have access. Karen, the access for the class is for 1 year. It's a renewable license. And again, you get access to all the material for a whole year. You can watch the videos as many times as you want. All of the webinars are recorded, and so if you miss a live webinar, you get access to that webinar after the fact in the mastery class private group. So thanks so much for coming today and listening, and I hope to see you around the field of flow. And again, if you have any other questions, expert@expertcytometry.com is our email address. And this last couple of seconds, the mastery.expertcytometry.com to get that special bonus. Thank you so much. Have a great day. And I hope it's warmer than where it is here in Rochester with our snow. Oops.